Welcome to another chapter in the saga of evolution. So today we're going to try to discuss the evolution, how it occurs in populations. Before we have talked about the evidence that we had for evolution, and we mentioned we had evidence from the fossil record, we have evidence from anatomy, homologous structures, vestigial structures, and embryology. We had evidence from the molecular domain with the sequences of amino acids and nucleotides, and we also had all the evidence that we got from natural selection and artificial selection, basically that organisms are changing over time, and we can study and document that change. Basically, the main thing is that we are seeing it. Evolution is still happening today. It happens constantly. You cannot stop it. It's always going on. And it's just because evolution basically means that organisms change over time, and they are changing in response to the environment. That's the premise. So we are going to start with evolution of populations, because even though we have individuals, the individual doesn't matter at all, because you're going to die. What's important is what you leave here for the next generation. And remember that only a few individuals, the ones with the best adaptations, are the ones that are able to reproduce and leave something for the future. So not all of us are going to count, and not all of us are going to contribute for the future generations. Now, please remember that humans, because we control our environment, we change the rules of the game. So there is strong evidence that humans, we are still evolving in some aspects, but we are changing a lot of the rules, and we are controlling how we do it. So please try not to think too much of humans from now on. Up to now, we have been subject to the laws of nature and evolution, but in the last 100 years or 200 years, since we started controlling with the development of technology, or with the progress in technology, we are bending the rules. But it applies for everybody else pretty much. So, important concept here is that natural selection is going to act on an individual. You are going to get to survive or not. And if you survive, you pass on the genes. If you don't survive, that's the end of all your genetic endowment. But what is going to change, and you're going to see the change, is in the next generation when you look at all the organisms together. So that's an important concept that we have, that natural selection is going to act on individuals, but is the populations, really, the group of organisms that we are going to see as evolving, the genetic makeup of this population. So the other point that I want to make here is that we are so focused sometimes in just physical traits, that we want to see changes in the physical environment or physical makeup of an organism, that we forget that we are basically just a bag of chemical reactions inside. And sometimes we really need to look at those. And so change is not only in the physical aspect, but change is also in our genetic composition, biochemical composition inside. And just to bring that aspect here, I have this graph here that evolution is not necessarily physical traits, but can also be a biochemical trait that you cannot see necessarily immediately. So this is a nice fish, the mamichog. They are common along the coast, Atlantic coast of the United States. And they go from 44 degrees latitude, far north, Boston and the like, to 30 degrees past south of Virginia, North Carolina, South Carolina. If you look at the distribution, if you look at it, they have a particular allele, lactate dehydrogenase. This is an enzyme that obviously removes hydrogen from lactate. All right. If you see the distribution of this enzyme, they all have it, obviously, but there are some forms of this enzyme that work better at low temperatures, and some forms of this enzyme, so different shapes, that work better at higher temperatures. So low temperature better, higher temperature better. Still work, but they have obviously a different shape. They are not equally responsive. So if you look at the frequency of the cold-adapted allele, basically the enzyme that works better under cold conditions, colder waters, notice where you find it. If you examine all these populations, in populations up north, 44 degrees of latitude, pretty much all the alleles, almost 100%, frequency of 1, almost all the alleles are the cold-adapted one. Obviously, it makes perfect sense. If you have a fish with the warm allele here, it's not going to work very well, very slow. However, if you go down to the lower latitudes, 32 degrees, guess what happens to the frequency of this cold allele? It's pretty much gone. So here, who has higher frequency? Obviously, the warm adapted allele. So the point here is I want you to realize that it's not only the physical, what we see on the outside. Sometimes the important things that give an individual an edge is at the biochemical level. So always, 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 please look at the graph, read carefully what it says on all the axes, and try to understand the data. It's very important that you do that in this unit. It's all data. So changes in the population, we know populations change. We already talked about how they are changing the makeup of insects and bacteria, that because of the selective pressure of insecticides and antibiotics. And now I want to show you another one. And this is again experimental data in the wild. This is regarding guppies, and a lot of you know guppies is a fish that you can find in tanks. And if you have fishes in tanks, you know guppies are very colorful. The females are kind of dull, boring, but the males have beautiful tails, a lot of different colors. That's what we call sexual dimorphism. Males and females are different. We are sexually dimorphic. So in the wild, you know, these fishes that we have in tanks all come from real places in the wild. There are other fishes that you can find in the lakes where you find guppies. And they found a system, actually, this is why it's so wonderful. They found a system of 2 lakes. And the lakes are connected, so the fishes can actually move there through a river. But in one of the lakes, you only find guppies and you find killifish, that lake. In another lake, connected to the first one, you have the guppies, you have the killifish, but you also have pikes. If you look at the color patterns of the guppies, in this lake, where you only have guppies and killifish, all the guppies are bright, colorful, beautiful, just like what you see in the tanks. If you go to this lake, where you also have pikes, a top predator that eats guppies, look at what happens to the pattern of coloration in the body of the guppies. They're all dull, boring, you can barely see them in the water. So, what's happening here? Same species, the populations are connected so fish can move, but when you look at this lake, beautiful, brightly colored fish, what happens here? All dull. Why is that? What's happening? This pretty much has total variation. What happens here? What happens to you here if you are brightly colored? You are going to be eaten in a jiffy by the pike. So, it's not that here you don't produce brightly colored fish. What happens here is that all those brightly colored fish are eaten really fast. Do they get to reproduce? No. And if they don't get to reproduce, the pattern of coloration starts getting lost over time, because only those really boring, dull fishes are the ones that have the edge and get to reproduce. So over time, these two are going to become very, 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 very different, because the selection pressures are totally different. Alright, so populations changes and the data to prove that. Of course, I'm telling you a nice story, but without the data, it's kind of purposeless. So, places with high predation. You can count actually the number of spots per fish. Places with high predation, very low number of spots, pre-dull. Places with no predation or low levels of predation, you get to see the nice, beautiful fishes because it's selective survival. So the data is there to support what the theory kind of tells you. So again, please take into account we have data to support all these things. Alright. So what happens here? So remember again, individuals are the ones that survive. Individuals are the ones that get to reproduce, of course. Individuals are the ones that are selected. I have the good genes, I get to reproduce. But we see the population as a whole evolving because I'm gonna die. I'm out of here. It's all my offspring and the interactions of my offspring with the other offspring that were lucky enough to survive that are gonna make a difference. So populations evolve. So now I want to show you a concept of fitness. Fitness basically is a measure of the reproductive success of an organism. So if I get to reproduce and pass my genes on, I have high fitness. If I don't get to pass my genes on to the next generation, I have low fitness. So again, individuals with one phenotype obviously are going to leave more offspring. Again, I want to bring your attention to the data, and I want you to analyze this carefully. You have these, if you cannot see them, they are in your textbook. These are water striders, you know them, and we are going to analyze fitness. You know, fitness is a good indicator and a good thing. So number of eggs laid per day versus the length, the size of the creature. You can see that the larger the creature, the more eggs that organism lay per day. So if I asked you who has the greatest fitness here, you would say obviously the larger individuals because they release or they lay more eggs. Good. However, life is not that simple. If now we look again length of the adults, but now instead of number of eggs, we look at the lifespan, how long these water striders live, what do you notice? Smaller ones live on average 30 days. The really large ones, how long do they live? They are going down to almost 20 days. So you might be large and lay a lot of eggs, but you are large, you don't live that long. So if you mix those two things, how many eggs you can lay and how long you live, you come up with a very different graph for fitness. Length of adults, just like before, but here is the number of eggs laid during their lifetime, not per day, during their lifetime. And what do you see here? Who has the greatest fitness? In this graph was here, but we know that's wrong because we need to take into account lifespan. Obviously, the greatest fitness belongs to individuals that are about 14 millimeters long. Just to keep you thinking about the concept of fitness, that you always have to dig deeper than the surface. So, let's look at variation. That's what we are talking about. There is variation. Some variation is good, gives you an edge, is an adaptation that allows you to survive, and you get to pass on that to the next generation. Now, where does all this variation come from? Where does the variation come from? Because we see variation. Everything is different. Even things that kind of look the same, when you look at them closely, they are different. What's the ultimate source for all variation? How are we truly different? All variation, please, 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 the only source, the true source of variation is mutation. Random changes in our DNA that get to be passed on to future generations. please, 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 all variation comes from mutation. Now, the other things that we do, like sex and meiosis in particular, basically all that it does is shuffles the genes. So here is where novelties arise, new genes, new things, and then anything related with sex, including meiosis, basically what that does is just shuffle the genes and make different combinations. But mutation is the source of new or novelty in organisms. Okay? So sex allows for recombination during meiosis, new combinations, new phenotypes, and then if you have males and females, even better, because then you have extra combinations that you can make. So this, I recommend that you write it down, make a nice chart to organize your thoughts, because this is extremely important now. What sort of changes are going to cause evolutionary change? What sort of things favor these changes in organisms? And there are actually 5. All the things that happen, we can group them into 5. The first agent of evolutionary change, obviously, is mutations, basically chemical changes in the structure of our genetic material. Second source of evolutionary change in populations, remember we are talking now populations, is the concept of gene flow. Genes flowing from one place to another, genes moving. That's basically the concept of migration. Non-random mating. When you get to choose who you mate with, you are selecting. This is sexual selection. Please take note. Next one, genetic drift. This is a new concept for you. And genetic drift is changes that are going to happen by random events. Has nothing to do with adaptations, has nothing to do with being the best. It's just you got lucky. And the last one is selection, true selection, which makes sense. So please, you need to know this in your sleep. What are the 5 agents of evolutionary change? Mutation, gene flow, non-random mating, genetic drift, pure chance, and selection. And now I want to go through each one of them very quickly to give you an idea of what each one of them represent. So we start with mutation. Again, we know this, variation, and of course, because DNA changes, ultraviolet radiation, mutagens, different chemicals in the environment, just pure chance we have. And mutations are happening constantly. Now, if it's a good mutation, gives you an edge, get selected for. If it's a bad mutation and it has low survival rate, it disappears pretty fast. And remember that there are mutations that are silent because they don't affect the ultimate product, which are the proteins. So keep that in mind. Now, every time you have a change in a mutation, if you happen to have a change in the protein, remember, if it changes the sequence of amino acids, because we have polar and non-polar amino acids, it's going to change the folding pattern, it's going to change the structure. This would be a good time for you to go back and review protein structure, primary, secondary, tertiary, and quaternary, and seeing how changes in the amino acid sequence affects the different levels of protein structure. So, mutation, agents, check. Next, gene flow. This is a tricky one because for some reason seems to be one that causes troubles to students, so beware. So, oh no, not gene flow, sorry, I'm skipping up. Gene flow is a piece of cake. It's the movement of genes between one population or of another. So basically, if you are an animal, you move from one place to another. Wherever you go, you are going to bring your genes, and if you end up mating wherever you go, you are spreading your genes. No problem. Now, do plants migrate? Good point. They don't move around, they don't get up and walk, but remember, plants reproduce using pollen. Pollen basically is the male gametes that can fly. So even though the plants cannot move around, they can spread their gametes through the air, Superman style, and they can basically introduce new genes to other populations if they can get carried away. So what's this migration, this gene flow gonna do? Well, you're gonna start mixing genes across different regions, and it can result in two processes, very different. Basically, by mixing it, you can reduce the differences. So populations that are very different, when you start mixing their genes, they become more homogeneous, kind of an in-between type of thing, because they are sharing their genes. So that's one thing, reduce differences. But at the same time, another thing that gene flow can do is it can introduce new alleles into a population, bring things that, if I'm a population here, we are a population here, and we have a visitor from outer space, it's going to bring things that we don't have here. So it's going to introduce new alleles. So please beware the two very drastic effects. It can make you more homogeneous as a population, or can introduce new alleles. Very different outcomes. And for this we have humans. Up to about 200 years ago, human populations were clearly distinct in the different parts of the world because we had very low mobility, pretty much nothing. However, with the advent of technology and transportation improvements, now we fly everywhere, we live everywhere, and we basically, this is what we are becoming. As long as we keep on increasing and maintaining the gene flow that we have now, this might be the new face of America, a blend of everything that we have here. And we can see that happening already. Time to think. So, mutation, source of variation. We have gene flow, source of changes. Next one is the concept of non-random mating. So mating is not random. Now, if you have, let's back up a minute and let's go to just quite the opposite, and let's do random mating. Random. What does random mean? Random means that, I'm gonna use the example here. I have, let's pretend I have a sea urchin. You know, sea urchins. Sea urchin in the ocean. I have a female sea urchin here, and I have a nice male sea urchin there. That's a male, female, and male. How these guys move very slowly. If you don't like sea urchins, think about any of the creatures that are sessile, that are always stuck in the rocks, that cannot move very much. Corals, you name it. Think about all those creatures in the ocean, please, that are sessile, that do not move. So what happens here? When reproductive time comes around, the female sea urchin releases the beautiful eggs all over the place into the water. You can see it actually. There are awesome YouTube videos showing the release of the eggs by female. Together with the eggs, of course, they release hormones into the water, signals, and these hormones are detected by the males. Once the male detects the hormones, and that's the signal, oh my God, the female just released the eggs, what do the males have to do? The males are gonna release their sperm. And all this is happening in the water. Egg, sperm, pure chance who mates with what? This is totally, for the most part, random mating. Because you don't know which egg is gonna find which sperm, or which sperm is gonna find the egg. So most of the creatures, a lot of the time, a lot of the creatures in the ocean, even plants with the pollen flying around, pollen flying around with wind, is totally random mating. Random is good, no preference. That doesn't cause change because it's totally random, even mixing, no preference. So, this is not going to cause change. Oh, by the way, next time you go to the beach and drink water, you know, you are swimming and you swallow some water, just think about what sort of things are in that water. So, that was random mating, just eggs and sperm randomly, and they find each other, and just fair game for everybody. However, we know for many species the mating is not random. There is what we call sexual selection, when there is a preference, when you choose exactly who is going to fertilize your eggs. And usually the female is the one that chooses who you want to fertilize your eggs. So in most cases that we have this non-random mating, that you, of all the choices, it's not random, no, I want you are limiting the options for sperm. This is sexual selection, and a clear indication of that is usually some type of sexual dimorphism, where males and females look different. And you are familiar with lions' manes, with birds' colorful males, and with mammals like deer, reindeer, and stuff like that, that the males are the ones with the huge antlers. Frogs, the males are the ones that sing at night. So, sexual selection, this is a beautiful concept. Is once you know about it and you start looking around, it's fascinating, even for humans actually. So in this species, when you have this, the female is the one that is going to make a choice, and the female has to judge around, okay, which male has the best genes, because I want the best for my offspring, so I'm going to try to choose the male with the best genes. Now females are going to choose males based on different characteristics depending on the species. If you are a frog, the frog with the biggest, most powerful sound, the male with the most, is the one that is going to be preferred by the female frogs, obviously. A wimpy croak is like, that's a wimp, I don't want those genes, I want the powerful ones. Same thing if you think of birds and mammals. Females are always choosing the ones with the bigger antlers, the one with the more brightful colors, the ones that are the most expressive in their mating dances, like the blue-footed boobies. You should YouTube their mating dances, they are beautiful. In all this, the female is choosing the male with the loudest voice, the brightest colors, the biggest antlers, always the biggest mane. If you're a lion, is fascinating. Look at those two, the male with the biggest feathers. Now, what are those traits telling the female? It's not just that because you are the biggest or the brightest, you are the best. Best, not a good word, but still. All comes back to this beautiful concept called the handicapped principle. What is the handicap principle? The handicap principle, and I need you to think about it, is like, I am one of these colorful birds, okay? If I'm the most colorful, you're gonna select me, but so is my predators. If I'm this bird in the forest with bright colors, I'm screaming, hey, I'm here, eat me. Now, if despite the fact that I'm calling attention to my predators, I'm still alive, that means I'm good. That means I have other genes in me that, despite this handicap of being alerting all my predators, I'm still alive and survive. So as a package, having these handicapped genes that attract predators are also telling me, hey, all my other genes must be really good for me to be still alive, and I'm gonna be a good father to your offspring. Think, for example, of the deer. These antlers weigh a ton. Not a ton, but a lot. You have to have strong neck muscles to carry them around. And you live in the forest with branches. These things are getting stuck everywhere where you eat. Despite all that, you are still alive. You must be really good. And the females want that. So think about it. It's beautiful. Now, all this is kind of anecdotal, but we have data to show that this is truly that when the females choose these weird traits, it's really an indication of something else going behind. And I want to show you the data for the lion's mane. Where do lions live? In Africa. Hot! And you have this huge mane in the middle of the summer there, or all summer all the time. Hot, hot, hot! You cannot be happy having this huge mane in a hot place. The females are smart, they don't have the manes, but the males do. So you say, gosh, that huge mane, poor lions, they have to thermoregulate even more than regular non-mane creatures because they are overheating all the time. That puts a heavy toll in their physiology because they have to maintain the proper body temperature, thermoregulation. So what did they do? They looked at a number of parameters. Mane, for example, over here is the age of the lion, and they did a correlation between the mane and, for example, here, amount of testosterone. The bigger the mane, the most testosterone. Length and age. The older you are, the more mane. And darkness, the older you are, the darker your mane. Hey, if you are old, that means you have survived all this time. So the most important one, among other things, is the correlation with testosterone. That actually having a bigger mane correlates beautifully with higher levels of testosterone in the lion's body. And when you look at the testosterone levels, testosterone actually increases the size of your muscles. That means that you're bigger, you're faster, you're stronger, you can catch more prey. You are more aggressive. That means that you're also going to be able to protect your females from all the intruders. That means also that you can produce more sperm and you have higher fertility rates. So if I'm a female, I want a male that is going to do it and be efficient and get me pregnant. I don't want to be messing around with a male that doesn't accomplish that. Also, the amount of testosterone correlates with longer life. So testosterone, beautiful indicator of all the good things that I want for my offspring, and the one external feature that allows me to kind of judge all this full package is actually the mane. So the mane is a kind of a true indicator of the presence of all these other beautiful traits that are important for survival of my offspring. So, who am I going to pick if I'm a female out there? I'm going to pick the male with the biggest mane to fertilize me. Because despite being extremely hot, the poor guy is going to burn and have to thermoregulate more. Hey, the whole package is beautiful. Gives me great genes for my offspring. Think about it for all organisms. You have this male frog croaking in the middle of a pond. Ribbit, ribbit! And who do females prefer? The loudest one. Because despite being advertising, hey, I'm croaking, I'm here, come and eat me, is able to survive. That means it has to have a package of genes that are excellent. And I want those for my offspring. I hope you got this. It's cool. So, what agents of change have we looked at so far? We had mutation, we have gene flow, movement of genes among populations, and you have non-random selection that is going to allow to move in a particular direction. Now comes the one that I got messed up at the beginning, that genetic drift. Genetic drift. This is when the frequency of certain genes changes, but it's because it's totally random. It's not because you are well adapted, it's not because of anything, it's just you got lucky. Pure, absolute luck. So it's not related to adaptation and it's not related to selection whatsoever. Pure chance. Look at that picture there. Population of beetles and just by pure chance, 2 of the green ones get squashed. Pure chance, nothing else. Because nobody's looking to eat them or anything. The remaining population has a completely different genetic makeup because of pure chance. This is genetic drift. That is genetic drift. Now, there are 2 kind of examples of genetic drift that are very cool, but please remember these 2 are examples of drift. And one of the examples is what happens with what's called the founder effect. And the picture is better than nothing. You have a population, mainland for example, yellow and red, huge diversity. Founder effect is like in the case of Galapagos or any other territory that organisms are going to colonize. Think of right after volcanic eruptions, think of new islands like Hawaii that just pop underground through volcanoes. Of this original population, who are the ones that were able to survive the trip just by chance because they got lost in a storm or something that made it to the island? Obviously, only the red one. So, what happens with the new population that you have now colonizing this new area? Is it a true representation of the original population? No, because just by pure chance, this is the one that made it, so now the new population looks different. This is called the founder effect. Depends on who founds it, is the makeup of the next generation. Small group splinters off and starts a new colony, and it might not be representative of what you had there. That's one special case of genetic drift. Another one is called the bottleneck effect. And what is the bottleneck? The bottleneck basically is you have some factor of disaster, a natural process that reduces your population, not necessarily because of who is better adapted or anything, just brutal changes that destroy most of the population, and you only have a small contingent alive. Just by chance, that small contingent gets to repopulate the next generation and might not be a representative sample of what was there before. And I'll give you a couple of examples in a minute. So, founder effect example here. We have humans. You are familiar with this, the out of Africa hypothesis. Humans emigrated from Africa and through the years colonized the whole world. Human populations, I think this is fascinating. This is the distribution of type O blood in native populations. So before we started all this traveling and mixing, blending into each other. If you look at type blood O, blue means that 90 to 100% are type O. So pretty much 90 to 100% of the population here in America is type O. If you want to know what the darker red, 80 to 90% type O. So overall, if you look at America as a continent, what type of blood is the dominant? Pretty much 90% of the population here is type O. Why? How do you explain that? Where do people came to America? Who are the people that colonized America? People that were able to cross the Bering Strait. Obviously, the people that crossed here 20,000 years ago were pretty much a small band of humans that had type O blood, and they are the ones that delivered that gene through the whole America. The rest of the world is a mix of the percentage of O is much lower. But in America, extremely high because of the founder effect. That band of humans that came were O. That's beautiful. Absolutely fascinating. So, I told you drift, these are changes just by chance. We had the founder effect, just by chance. And the other example I wanted to show you is the bottleneck. You know, bottleneck. So, best example as always are drawings, and because drawings you can see the stuff. So bottleneck is when you have a large population and just by random, has nothing to do with adaptation selection, most of the population gets destroyed. Usually it's because of a natural disaster or something environmental, and then what you have left is a very small group of individuals with a very small group of genes, because everything else, the diversity, was wiped out. Famine is another one of those terrible factors. So look at this bottle. That's why it's called the bottleneck. You have a bottle with blue, green, blue, yellow, and red beads representing genes in a population. That's your parent population. Then, because of a bottleneck, basically the diversity gets reduced and only a few get to survive. It's a drastic reduction, most of them die. Famine, natural disaster, lots of habitat, volcanic eruption, tsunami. Hey, there's nothing that can survive a tsunami there. So here is what you have left of all this humongous population. Now when these individuals, notice here, when these individuals, mostly blue and only 2 yellows, get to reproduce, obviously the next generation is going to be mostly blue, a few yellows, and you have one that got lost completely, the red one. So founder effect and bottleneck effect result in the same thing, that the next generation is different, but it's all due to a very random event that restricted the gene pool. So this loss of variation is just a chance. I want to emphasize that because this is where everybody gets tripped. And a lot of times you lose total loss of alleles, traits that are totally gone, like these red beads, and has nothing to do with fitness. Again, I pointed out this video is an 8-minute YouTube video, painful mating and the evolution of felines. If you are a cat person, watch it. But cats went through a significant bottleneck, and you're gonna learn something about that in this wonderful video. So bottleneck and felines with the last glaciation. Why do we care about this? And this is, think of this as a parenthesis, make a little parenthesis in your brain. Why these bottlenecks and things like that are important for us now that we are gonna have issues with conservation, try to help species that we put on the brink of extinction. Well, if you give you 2 examples, one man-made, another one natural-made cause of bottleneck. The California condor. The California condor pretty much almost extinct. This graph shows the number of wild and captive California condors from 81 till 2011. In 81 and 88, we had about 60 to 70 condors left. 60 to 70 individual condors, California condors, left because they had been eliminated by humans, and only human fault. Got it? Now the population even got lower here to about 30 condors. This is 61, 66. Here we are in the 80. Notice, probably 30 condors left. 30 condors left. So we started the process of trying to bring them back, breeding in captivity programs, heavily protection, endangered list species, all that stuff. And from then up to now, 2011, right now there are about 400 condors alive. They were listed as an endangered species in 66. So we brought back the condors. However, how is their genetic diversity? All the genes that you see here are exactly the same genes that were present here in the 30 condors that were left. Can you imagine how many genes have been lost? So if you look at the genetic variability of these species, is very, very reduced. And you know very well that when you have low genetic variability, you are very basically narrow-minded in that sense, that you don't have too many tools to deal with environmental change. So yes, we are bringing them back from extinction, but most of the genetic variability has been lost. And it's going to take thousands of years of random mutations to increase that genetic variability. We can do all sorts of things, cross them with Northern California, Southern California, but we're still playing with the same gene pool. Only mutation will increase their variability over the next millions of years if we survive. Right? So the other example that we have about this problem of bottlenecks is the cheetah. With the last glaciation event, the same thing happened to the cheetah, but naturally. So cheetahs have very low genetic variability because their populations went through a bottleneck during the last glaciation event. But they have recovered, and the last glaciation was 15,000 years ago, 10,000. So they are slowly going back with mutation rates increasing, but still very low variation. So it can be natural, like in the cheetah, or like in the condor and some of the other endangered species because of human activities, human trigger. So, last piece of change, we have mutation, we have gene flow, non-random matings, we have genetic drift, totally random events, and the last, of course, is natural selection, which is the king and queen of all the processes. You have the same beetles that I had before for drift here, but in this case, it's just a random shoe killing some of them. What's the selective agent here? The selective agent is the bird eating particular types, in this case eating all the greens. So natural selection, driving force in changing the makeup, because only those survive. Now, when you have natural selection, actually you can have different outcomes, and you can see what kind of outcomes you're going to have. So when you have selection, you know your population is going to change, but it's going to change at least in 3 different ways, and we have named these. So please focus on the very first one here. So notice here, this is what happens before selection, what your population looks like before selection and after selection. This is how is basically a frequency curve. I have few individuals like this, few individuals like this, most of individuals like this. And the spread is basically the range of a particular trait. If you want to use my example here, this can be very short people, very tall people. Most of the people are in the average size. So just so you understand how these curves are structured. Typical bell-shaped curve. Now, look at this one. What do you notice has happened to this trait, whatever this trait is? As you notice, the trait has shifted in the range of sizes, or whatever the trait is. The curve has shifted. This is what we call directional selection, where a trait is changing in a particular direction. And examples of this is the giraffe, the size of the neck of the giraffe. Over time, through evolution, just like the size of the horse, has been increasing. Why? Well, if you are a giraffe and you have a tall neck, that means you can eat leaves way above everybody else. And you have pretty much a very bushy, nice set of leaves to eat that nobody else has access to. That means I get to eat a lot, I get to increase my body fat a lot, and if I'm a female, I can have a lot of babies because I have a lot of food. So the next generation is going to be made mostly of larger ones, because the ones with the longer neck got to eat more, got to have more babies. So over time, you keep on favoring that. So that's directional selection, and 2 of the examples is the shift in size of the giraffe neck that neck of the giraffe that keeps on getting bigger, and the size of the horse. If you look at this next one, you will notice what's happening to the wide range of a trait. The trait is getting narrower. So the extremes, the really short and the really tall, are getting lost. This is a very interesting one because this has been seen, or is still seen, with the human birth weight. Over time, there is selection towards an average narrowed range of birth weight. And this is in places without medical facilities where birthing is a natural process, without all the high-tech stuff that we have these days. What's happening? If you don't have any of the technology to make the baby survive like we have in the developed countries, if you are in the third world or way out there, what happens? What happens to all the babies that are very small, that are born preemies or very small weight? They don't get to survive. So immediately from the gene pool, if you have genes that tend you to favor the small sizes, you are out of the gene pool because you do not make it. You do not survive. In technological developed countries, we have all the technology to make them survive, but in the nature, no. So we lose all those. Now, what happens with the really big ones, the really big babies? Big babies, big heads, they get stuck in the birth canal. And if you get stuck in the birth canal, you die and your mother dies. And you are out of the gene pool. So, in nature, humans, through evolution, this curve has been narrowed to a very narrow range from like 5 to 9 pounds. That's it. Most of the births are in that range in natural situations. Now, if you are in a developed country, what happens if you have a big head or you are a 10, 11 pound baby and you don't fit through the birth canal? C-section, and we get you out through the uterus in a different way. So we can bypass that. So we are messing around a little bit with human evolution, but birth weight has been restricted by the size of the head and the size of the pelvic structure that you have to go through, and by the inability of very small babies to survive. Stabilizing selection, because it gets stabilized in a narrow range. What's the other type of selection that we can see in nature? It's called disruptive selection. Normal birth curve, and what happens to this one? The average phenotypes disappear, and only the extreme phenotypes get favored. And a good example of this is what happens with mice that live in the desert. Actually, we have a case here in Arizona. We have light colored mice and black mice. Now, in the desert, if you are in the open flat areas, that's light colored ground. Obviously, the light ones get selected for because they camouflage. But in the desert, there are also rock formations, and most of these rocks are igneous rocks and other type of rocks that are dark. So you have a mix of light colored ground next to a dark colored rock. Now, who gets to survive in the dark colored rock? The dark one. So you survive either if you are a clear one because you camouflage in the lighter colored substrate, or if you are a dark one because you camouflage around the rocks. What if you are in between color? You are wishy-washy and you stand out in either one of those habitats, and that's where owls and eagles and all those other predators out there in the desert get you. So disruptive selection, we see that in nature. So the three types of selection. So again, we had just covered all the agents that cause change in populations. Mutation, gene flow, the movement of genes, sexual selection or non-random mating, because you are picking who's going to father your offspring. Genetic drift, sorry, that's my fourth, genetic drift, totally random changes, and the last one was selection. So these are agents of evolutionary change, things that change that. Now, why is good that we know that these 5 things are going to change evolution? Because now we can measure this. We can measure this. And that is what we are going to do later in one of the labs and in class. And it's called the Hardy-Weinberg principle or Hardy-Weinberg equilibrium. So, I want you to think about this. If you have none of these 5 agents of evolution, no mutation, no gene flow, no genetic drift, no selection, and matings are random, totally random, then you should not have any evolution. You should have no change. And from generation to generation, everybody should be exactly the same, because nobody's been preferred over other ones. This is what the Hardy-Weinberg principle basically established, that if there is no drift, no gene flow, no mutation, matings are random, and no selection, then the population is not evolving, and the gene frequencies should stay the same from generation to generation. Now, there is a mathematical component to this that we are gonna explore and study, and we'll do that in class, and we'll use the mathematical approach to figure out if populations are changing or not. Good? So, important that you know this in your sleep. If none of this happen, there should not be any change. But if any of this is happening, then you have change. Let me go back to the drift, because again, this is the one that causes most problem. Remember that drift is associated with random events that change the makeup of a population. Could be founder effect, could be bottlenecks, could be other factors, but are always because your population is very small. So how can you avoid this? If you have very large populations, any random effect is going to be minimized. So the secret to avoid genetic drift is always having large populations, very large populations, which doesn't happen with founder effect or bottlenecks. Just want to finish up with rounding up with a couple of 2 very specific examples of how some things are not going to according to plan. If you know, Tay-Sachs is a recessive disease. It's a lysosomal storage where you have a defective enzyme that does not break down lipids, and lipids accumulate in the lysosomes, and basically by 5 years old your brain is totally destroyed. Tay-Sachs was pretty much originally just found in the Jew population and some Cajuns, and now is spread through the population. Why? Because of migration, the mixing up of the genes. Now, since it has been it's a recessive trait, question is, where do new cases of Tay-Sachs arise from? If they die by the age of 5, where basically nobody gets to reproduce, so where are they coming from? Yes, that recessive. If a trait is not advantageous but is recessive, it can be hidden in the population forever and ever and ever until it shows up. Here is another case that I want you to be good at, and this is called heterozygous advantage. This is a particular type of selection where the heterozygous individuals have the edge, have the advantage over everybody else, and this is the example of sickle cell anemia. As you know, we have normal hemoglobin that produces normal cells, the hemoglobin normal. And we also have what we call the sickle hemoglobin. That's how we represent the allele. And that's the one that causes cells to sickle. Now, if you are homozygous for sickle, you will get sickle. If you are homozygous for the normal ones, you have the normal ones. Now, if you are heterozygous, they are normal, but under certain conditions of low oxygen, basically they sickle and change shape. So you are kind of in between. Now, if you are sickle, this is lethal, and basically you die by the age of reproduction. So you would think, okay, if the sickle is a bad one, then probably selection is towards mostly normal alleles. However, when you go to Africa, what's the main place where you find this, and you look at the population, you realize actually most individuals are heterozygous, not homozygous dominant, not normal hemoglobin. They have the normal sickle hemoglobin gene. And that was a puzzle. Why were the heterozygous being favored and not the homozygous dominant? And so that was a puzzle until they realized that there was a very interesting selective pressure here. Malaria is transmitted by the mosquito. This is a human here, that's the liver. The life cycle is the mosquito bites you, injects the parasite goes to your liver, reproduces or lives in the liver for a while, and then another stage of the parasite colonizes your red blood cells. And then it reproduces there in your red blood cells, and to get out of your red blood cells, they have to blow up your red blood cells, and that's when you get the symptoms of sickle cell. So you have the mosquito injecting, then the larval stages go to the liver, and then you have the parasite here inside the red blood cells, and they have to blow up to get out and be picked up again by a mosquito. So what's happening here? Well, the parasites do not survive very well in sickle cells at all. Sickle cells, among other things, have lower concentrations of potassium, and the parasite needs the higher concentrations of potassium found in the normal cells. The other factor is normal cells carry more oxygen. Sickle cells cannot carry very much oxygen. So the parasites avoid sickle cells at all cost. So, if you are a homozygous individual for normal cells, you are prime material for this parasite, and you get bombarded, and you die. If you are heterozygous, where you have some normal and some sickle, since the parasite cannot do well in the sickles, you have lower frequency of malaria in heterozygous individuals. Basically, being heterozygous gives you an advantage. So to summarize, if you are sickle, homozygotes for sickle, you die before reproductive age. You are out of the gene pool. If you are homozygous for normal hemoglobin, you are heaven for these parasites. They love you. And so a lot of you are going to die. Now, if you are heterozygous, then the parasite doesn't like you so much, so you have an advantage and you get to survive. And that's how heterozygous becomes the most prevalent, and that's why the gene, even though it's a deleterious, lethal gene, keeps on perpetuating because it gives you an advantage over the parasite. That's it, simple. So nothing is what it looks like. There is always another component, and you always have to look for the ultimate reason for everything. So you go back to Africa, the places where you have the highest incidence of malaria is also where you have the higher concentrations of heterozygous individuals. Beautiful. I hope that helps. Please think again. You need to know these 5 agents of evolutionary change in your sleep, and be clear that if none of those agents is working, your populations should not change. And we should be able to quantify that change using the Hardy-Weinberg principle of equilibrium. I hope that helps. Please enjoy.